Hello everyone, my name is Samiksha Rai and the topic for my presentation is HR-TEM technique. In this presentation we will talk about TEM and after that we will move on to HR-TEM and then we will know the difference between them, and after that we will know the term phase contrast, and based on that there is principle of HR-TEM, its working and the image formation, and after that we will know the reason for using HR-TEM and finally its uses and applications. Starting off with brief background to TEM. TEM or transmission electron microscopy is a technique which is a reliable way to produce high resolution images which rely on the interaction of electrons with the atoms in the sample. TEM uses similar principles to conventional light microscopy, but electrons are transmitted through a sample to produce an image rather than light. The next topic is Introduction to HR-TEM. HR-TEM or High Resolution Transmission Electron Microscopy is a type of TEM that allows the imaging of the crystallography structure of a sample at an atomic level. With this high resolution, it is possible to image crystal structures, defects in the crystals, and individual atoms. It is possible to have direct information about the crystallographic structure of the materials from images. Now we will talk about the difference between HR-TEM and TEM. HR-TEM is identical to TEM except that the magnifications used are high enough to easily see the lattice spacing of inorganic materials. TEM gives us more morphological and textural information, whereas HR-TEM gives us the crystal structure of material. So when there is TEM that work at higher kilovolt, those are usually designed for HR-TEM operation and are used for material science where the contrast is rarely a problem and the crystal structure is crucial. Moving on to phase contrast. Phase contrast is a light microscopy technique used to enhance the contrast of images of transparent and colorless specimens. It enables visualization of cells and cell components that would be difficult to see using an ordinary light microscope. Phase contrast is used to visualize transparent specimens when high resolution is not required. Now, principle of HR-TEM. HR-TEM technique relies on a principle called phase contrast for image formation, which can often come with some problems when used at such a small scale. When the image is formed through the objective lens of the microscope, there is some significant oscillation present due to the small scaling used, which can often reduce the quality of the resolution in the image. A function called phase contrast transfer function describes how these limitations affect image formation. The transfer function is universal for all lenses and for all specimens used, so it can be easily measured in all scenarios and used to predict the resolution produced in certain procedures. The principle involved in TEM and HR-TEM is similar, but HR-TEM provides high-resolution images at atomic scale level. HR-TEM is a type of TEM but uses both the transmitted and scattered beams to create an interference image. It is a phase contrast image and can be as small as the unit cell of crystal. All electrons emerging from the specimen are combined at a point in the image plane. The picture is then finally seen on the screen, which is magnified by approximately 10 to the power 6 times. Now the image formation in HR-TEM technique. The beam of electrons from the electron gun is focused into a small thin coherent beam by the use of the condenser lens. This beam is restricted by the condenser aperture, which excludes high angle electrons. The beam then strikes the specimen and parts of it are transmitted depending upon the thickness and electron transparency of specimen. This transmitted portion is focused by the objective lens into an image on the phosphorus screen. Optional objective apertures can be used to enhance the contrast by blocking out high-angle diffracted electrons. The image that is then passed down the column through the intermediate and projector lens is enlarged all the way. As seen in figure 1, the image strikes the phosphorus screen and light is generated, allowing the user to see the image. The darker areas of the image represent those areas of the sample where the fewer electrons were transmitted, while the lighter areas represent those areas where more electrons were transmitted through. In the second figure shows a simple sketch of path of the beam of electrons from above the specimen and down the column to the phosphorus screen. As the electrons pass through the sample, they are scattered by electrostatic potentials set up by the constituent elements in the specimen. After passing through the specimen, they pass through the electromagnetic objective lens, which focuses all the electrons scattered from one point of the specimen into one point in the image plane. In the figure, we see a dotted line where the electrons were scattered in the same direction by the sample and are collected into a single point. This is the back focal plane of the objective lens and is where the diffraction pattern is formed. Now let's move on to the next topic of why HR-TEM. HR-TEM uses slightly different methodology to other types of TEM, resulting in high resolution that can structurally characterize samples at an atomic level. It is therefore a very useful technique in the study of all nanoscale structures. Structures at this scale normally are very difficult to visualize and characterize, so using this method opens up a whole new way to research into areas such as nanotechnology or material science. Now the uses of HR-TEM. HR-TEM is often used for the study of crystals, but also materials such as metals, semiconductors, and nanoparticles. Special focus is usually aimed at defects in these materials at the atomic level. These defects can include point faults, linear faults, or planar faults. This depends on if the structural defect affects just one exact point 1D along a line or 2D respectively. Point defects can often be corrected but can be harder to detect. Planar faults such as crystallographic shear planes, stacking faults, or interfaces can be a bigger problem in the quality control of material production and normally depend on specific atomic interactions within a material. Now on to the last topic, applications of HR-TEM. Imaging with HR-TEM enables individual atomic columns to be resolved in most inorganic materials, making it possible to determine the atomic scale microstructure of lattice defects and other inhomogeneities. Additional information can be extracted from high-resolution studies, including unique insights into the controlling influence of structural discontinuities on a range of physical and chemical processes. HR-TEM has impacted many scientific disciples, and the technique has generated a vast scientific literature, far too extensive to be reviewed in any detail. In the figure, we see in figure A, HR-TEM micrograph showing core structure of symmetrical edge dislocation, and in figure B, that is corresponding structural model.